Alors on continue avec cette fois, on est toujours dans des approches vraiment qui aujourd'hui sont encore fascinantes mais balbutiantes, mais qui ouvrent des horizons absolument incroyables. Là, on n'est pas forcément dans une révolution au niveau d'un grand champ de la recherche, mais dans un outil dont on ne fait plus que parler de lui depuis quelques années, dont on prédit au moins un prix Nobel sous peu, qui a été co-inventé d'ailleurs par une Française, et une américaine, et qui aujourd'hui est un incontournable de nombreux laboratoires. Je veux parler bien sûr du CRISPR-Cas. Et pour nous en parler, entre autres, mais notamment pour nous en parler, et plus globalement de la chirurgie génomique, j'appelle Fulvio Mavilio. Merci de nous rejoindre. Fulvio Mavilio, il est le directeur scientifique de Généthon, et donc il utilise bien sûr, il travaille énormément sur cette technologie qui a été développé et qui aujourd'hui est un outil majeur. You wanted to make a presentation in English.
Oui, bonjour à toutes et à tous. Je suis en France depuis 5 ans et mon français a progressé, mais pas vraiment pour vous donner une présentation sur des arguments un peu compliqués comme la chirurgie génomique.
Alors on vous écoute.
Merci. Je tiens à remercier d'abord les organisateurs pour me donner l'opportunité de présenter l'innovation d'Evry de notre côté, l'innovation sur la thérapie génique et notamment sur la genome surgery. Et pour souhaiter évidemment à Pierre qui est la 3e phase de ta vie professionnelle, l'après-vie, soit créative et formidable comme la première et la 2e. Merci pour tout ce que tu as fait pour Evry et pour l'innovation française, européenne et pour nous tous. And then I'm sorry, but I have to switch in English. What we have been working on for the last 25 years and the last 5 of my life in Généthon is gene therapy. Gene therapy is quite simply the use of genes as medicines. So there are many different forms of gene therapy. This works. And you can see 2 versions of this in this slide. And one that's called ex vivo, which essentially means using the cells of the patient, cells that are taken from the patients, genetically engineered by using vectors, which reminds me that we were sort of doing the first forms of synthetic biology in the 1980s when we were trying to transform natural viruses into strange animals that can carry therapeutic genes into patients. And another form is using the same kinds of animals, but injecting them directly into patients. I will concentrate the first minutes of my presentation on the first form of gene therapy. So essentially using stem cells modified by vectors with therapeutic genes as a form of therapy, as a form of genetically modified transplant. And this is essentially the scheme of how this works. You have a little patient here who, for instance, has a disease in his blood cells, a blood genetic disease. What we can do with gene therapy is taking cells out of his hematopoietic systems or the bone marrow, modify them with a vector that comes from retrovirus. Today we use something that's derived from the HIV virus, in fact, and then transplant them back into the patients. And of course, if this whole thing works, the therapeutic gene is here in red. The stem cells of the patients will be permanently genetically modified, and so the progeny of the cells will have this genetic modification, and hopefully they will be corrected and the patients will be cured. The first example of this came in the early 90s this is one of the things in which Europe essentially led the way for many, many years, and it's probably still leading. The application of gene therapy in clinical medicine. And the first diseases that were approached by this technology were severe combined immunodeficiencies, and this was work that was carried out at the same time in Italy when I was working there at that time, and in France. And that was successful, and we learned a lot of things out of gene therapy of immunodeficiencies. Just to show you that this was a long but successful path. This boy, you may know the story. It was one of the bubble boys. This was a boy that lived in the United States into a bubble because he didn't have an immune system, and there was no cure for these diseases for patients that do not have a suitable donor. So gene therapy essentially filled that type of vacuum and provided a cure, not for this particular boy that unfortunately died before these forms of therapy became available, but for many others. And severe combined immunodeficiency, these are sort of historic papers that in the mid-90s and at the end of the 90s essentially established through clinical trials the gene therapy as a form of medicine for these patients. And just to remind you that what we're talking about is really human beings. I like to show this very old movie that shows one of these first patients. This was a little girl that came from Colombia, and she didn't have an immune system. And thanks to the gene therapy technology, she could have a transplant of genetically modified stem cells. She doesn't look very happy here, but I can assure you this is a really normal person now. And Maria Grazia Roncarolo and Alessandro Aiuti were the brave clinicians that started this venture, as I said, in the 90s. Okay, let me go back to my presentation if I find it. Of course not. What is it? Lost forever. Ah! Help. C'est où ma présentation? Oui, voilà. Oui. It's funny because I spent 20 years with the Macintosh having problem in Congress centers that use PCs, and now I have a PC and I have to present on a Macintosh. Okay, so, but essentially, the reason why I'm telling you this story, not only because patients are cured, but because this is sort of where the story ends. And last year, the therapy for that you have just seen has been transformed by a pharmaceutical company, licensed and transformed by a pharmaceutical company into a commercial product. So this is the beginning of the commercial phase for gene therapy. And for those of us that have lived this story from the beginning, this is very good news. So the second example that I would like to make is another disease, and this is a disease where Généthon had a pivotal role in curing, and in particular Yang Ali, who is in the audience, who really, with her leadership, put together a group of clinicians that carried out a clinical trial that showed that this terrible genetic disease, I don't have the time to go into the details of it, but it's essentially an immunodeficiency with autoimmune problems, can be cured by gene therapy. And again, thanks to two clinical trials carried out at the same time in Italy, with Généthon in England, in France, at Necker in Paris, and the Boston Children's Hospital, at Harvard University in the United States. All these people collaborated in showing in a number of patients that this therapy is effective, that this disease can actually be treated by gene therapy. And with these two, again, papers published, the Italian trials and the Généthon-sponsored multinational trial that came to essentially the same conclusions. And also, I always like to show this picture, which is, again, real human beings. And this was the 2015 poster of the telethon, and essentially says, if you want to know, well, I don't have to translate this for you. That's ridiculous. Okay, so today with this technology, that there are a number of diseases that the scientists all over the world are addressing. Not only immunodeficiencies, but also other very complex and diseases like Fanconi anemia, like central nervous system diseases that can be treated with the same technology. And very importantly, and we spent some time on this, hemoglobinopathies. So thalassemia and sickle cell disease, which in France is called Trapano Cytosis, which is an important disease all over the world, but in particular for this country. Which brings me to the issue of gene editing. So the technology I showed you clearly works, clearly shows efficacy in treating fatal diseases. And it's sort of, at this point, prehistoric form of technology. We take a virus, we transform this virus into a vector. The vector carries the therapeutic gene instead of the gene of the virus. And this essentially is a way of transferring a piece of genetic information, a piece of gene, into the genome of a patient and correct, in a way, a genetic disease by providing an extra copy of a functional gene. So it's like you have a flat tire in your car, and instead of changing the tire, you add another one. So you have 5 tires, but this, of course, works. But can we think of essentially doing something better, which is not adding another piece of DNA to the genome, but just correcting the genome? So if we have a genetic defect, can we just change the bases that are responsible for this defect and put the genome back to where it should be and where it is in most of the population? So this is exactly what's called, what is gene editing. So gene editing is essentially the use of technology to change the genetic information of a cell, not by adding information, but just changing it. So by changing the bases, directly modifying the DNA sequence. This has been possible for a number of years, actually for almost 30 years. But in very inefficient ways. And as our moderator reminded us at the beginning, very recently this technology improved, and improved thanks to discoveries, really seminal discoveries from a number of people. And you see some of those in these slides. George Church in the United States, in Boston, Feng Zhang at the MIT, and Emmanuel Charpentier, who is French, and Jennifer Doudna, that actually essentially designed the first step of what we now know as CRISPR-Cas9 technology, which is a derivation of a study on the innate immune system of bacteria. So to go back to the discussion we had before, if we only work on things that we think are useful, we never discover that much. So this is sort of a serendipitous discovery done by people that were interested in how bacteria defend themselves. And essentially, the modified version of this bacterial protein is what we now use very efficiently to edit the human genome. So gene editing is trying to go fast on this. This gene editing essentially is based on a combination of proteins that we use to cut the DNA, and the machinery of the cells that the cells uses to repair the cut that we introduce. So it's a combination of natural and synthetic system. The synthetic part is that we use a protein, we will see what kind of proteins, to introduce a double-stranded cut into the human genome. What happens then? A cell tries to repair a double-strand cut. This is what our cells do every day. And it can repair it in two ways. It can repair it using one machinery, which is called non-homologous end joining, which essentially means taking the two pieces and bringing them back together. And this is a system that introduces also mistakes in the genome. We can use it, for instance, to create deletions. I will show you an example of that. Or to introduce mistakes, random mistakes, and for instance, kill genes, do gene knockout. But also we can use the cell, actually not we, can use another system. The cell has two systems to repair DNA, and this is called homologous recombination, which essentially means by using another template, for instance, the other chromosome, or something that we give to the cell as a template, the cell can actually recombine molecules, and we can fix a genetic mutation by this. If there is a genetic mutation here, we cut in close proximity, we use a template which doesn't have these mutations, and we correct the mutation. This works less well than this other system, but works, and will work more and more efficiently in the next very few years. So what will we use to cut? A number of proteins have been proposed in the last 10 years to do this targeted cutting of DNA. One is essentially all of this were proteins, protein modules that could recognize base pairs in the DNA. So protein modules that can recognize a certain sequence of DNA. If this is long enough, it appears only once in the DNA. So we can engineer proteins that go precisely in one place in our genome and cut it. There are several versions of this, and this is the way they look. So these are, again, proteins that we design that have amino acids that can recognize a certain sequences. And then there is a domain that essentially is a nuclease that cuts. So we can target these proteins pretty much everywhere. Protein engineering, though, is difficult. And this technology remained a sort of very difficult technology that very few people could actually master until the discovery of CRISPR-Cas9 came. So what is the difference? The difference is that CRISPR is a nuclease, like this part of these proteins, but does not use proteins to recognize sequences of DNA. It uses RNA. So it uses base pairings instead of protein to essentially target a sequence of the genome. Now, this may look to you like a small difference, but it's not. So here you see Cas9, and this is the protein, this is the DNA, and this is the guide RNA, it's called. So it's the molecule, the short piece of RNA that we use to target the protein anywhere we want. So there are two advantages for this. First, any student can design a piece of RNA to target a piece of any sequence in the genome. So it's very easy to use this technology. Second, the Cas9 is only one. So we use one reagent by just changing the guide RNA to target whatever sequence we want in the genome. No surprise that this now is becoming the favorite reagent to do gene editing. And as I said, any student and postdoc in any laboratory is using this technology to do all kinds of things. Can we use this for medicine? I will make two examples. One is research that is done in the US at this moment, and the other one is research that we are doing in Evry. So AIDS. You all know what is AIDS. So, and you all know that it's 25 years that we try to find ways of fighting the HIV virus with many things. Traditionally, by developing vaccines, or molecularly by developing drugs or other systems to kill the virus and the cells that carry it. The virus, the HIV targets cells by binding to a receptor, which is called CD4, but it also uses another molecule, which is a co-receptor, which is called CCR5. These two things are essential. If the cells doesn't have these two molecules, the HIV virus cannot get in. Now, we cannot remove CD4 from our lymphocytes because it's essentially for their functions. But actually, we can remove this co-receptor because there are patients, there are human beings, they are not patients, there are individuals that live perfectly without having it. So the idea is very simple. Can we actually knock out this gene by using the technology that I just showed you and create a population of cells that would be resistant to HIV virus? And this is essentially what people is doing in California, at the University of Southern California. And it's a clinical trial that essentially uses zinc fingers. So this was sort of, we can say, older technology with respect to CRISPR-Cas9 to cut into the sequence encoding CCR5 in the genome. And as I said, when you cut here, the non-homologous end joining introduces mistakes and essentially kills the gene. The result is cells that do not have the CCR5 co-receptor anymore. So these cells are taken out of the patients, they are expanded, they are transformed with the vector that carries these editing machines, and then they are given back to the patients with the hope of reconstituting in the patients a pool of cells that could never be infected by HIV. It is not a permanent cure, but it's a form of therapy because it's a form of, it's a way of providing patients with HIV-resistant cells. The second example that I want to make, and I will conclude my presentation here, is Trapano Cytosis. Again, you heard about this already this morning. This is a very serious disease. Actually, in Africa, it's endemic and it's catastrophic. The frequency of this disease is enormous. The number of kids that are dying every year for sickle cell disease is many more than those that are dying by all the wars that we see every night on television. And due to migration in the United States and in several European countries, and particularly in France, sickle cell disease is relatively frequent. There are almost at least 10,000 patients in France having this disease, and the frequency is 1 every 2,500 born every year. So births every year. So it's a serious disease, and it's due to one simple amino acid base change and amino acid change, base change in the genome and amino acid change in the hemoglobin, which is the protein that carries the oxygen. Now this changes catastrophic effects. The reason for this is that the hemoglobin chain that has this valine replacing this glutamic acid here polymerizes and creates sort of a molecular glue into the cell that transform our nice and flexible red cells into sort of this sickle-shaped red cells that are not flexible anymore, and they actually clog the veins. So what happens in these patients is that there is thrombocclusions, there are all kinds of ischemic episodes everywhere, organ damage, hemolytic anemia, stroke, all kinds of things, and severe pain, which is really a problem in this disease. So can we cure this disease by gene therapy? Well, of course, we can use a classical vector with the normal hemoglobin gene and try to synthesize normal hemoglobin, but clearly we need to remove the problem in the first place, and the problem is the mutation. So how can we do this? Interestingly, data that come from many years of clinical observation shows that fetal hemoglobin, which is the hemoglobin that we synthesize when we are really young, when we are fetuses, has an anti-sickling effect, so reverses sickling. So if in a red cells we have hemoglobin S, so the mutated hemoglobin, and fetal hemoglobin, what happens is that the more fetal hemoglobin we have, the more these polymers are reversed to normal hemoglobin molecule, and the cells returns to its normal shape. So there is a competition between hemoglobin S and hemoglobin F, fetal hemoglobin. So if we can, in a way, reactivate our fetal hemoglobin synthesis, we can, in a way, try to reverse the disease. And this is an experiment that nature has already done. And just in 3 words, what is fetal hemoglobin? When we are embryos and fetuses, we synthesize a different form of hemoglobin. This is because embryos and fetuses takes oxygen from the mother's blood. So the hemoglobin is a little different. And this is fetal hemoglobin. When we, around birth, there is something that happens that calls hemoglobin switching. So there is a switch, molecular switch. Fetal hemoglobin synthesis is shut down. Adult hemoglobin synthesis starts. But the genes are there. We just don't use them anymore. So there is a very complex molecular machinery that regulates this switch. Now, there are individuals that has a condition that's called hereditary persistence of fetal hemoglobin. So they are born, but they continue to synthesize hemoglobin F because of mutations. And it's interesting because a patient that has sickle cell disease and hereditary persistence of fetal hemoglobin does very well. So it has a very mild disease or doesn't have a disease for the reason that I showed you before. So we have clinical proof that if we can reactivate fetal hemoglobin synthesis in patients, we can cure the disease. How do we do it? Well, we can use gene editing to do this. And the reason is that we know that there are gene deletions in the hemoglobin. Now, this is complex. Try just to understand the concept. Some of these deletions we know cause thalassemia, which is another disease. And some of these deletions called hereditary persistence of fetal hemoglobin. Deletions in the locus that contains the hemoglobin genes. And it's deletion in particular of one region, which is the region that regulates the switch. So if this region disappears because of natural mutation, the switch doesn't happen, and the person is born and continues to make fetal hemoglobin. Now, can we destroy the switch by using gene editing and reactivate hemoglobin synthesis? So this is the type of work that we started 2 years ago at Généthon in collaboration with Imagine, the Necker people, and trying to do exactly this, to do gene editing to actually cut the sequence that regulates hemoglobin switch. So we use CRISPR-Cas9, and we deliver CRISPR-Cas9 to cells, not by complex viruses, which are difficult to manufacture and awfully expensive, but very simply by transfecting DNA. Simple DNA molecules that encode the Cas9 protein and the guide RNAs that guide this molecule to the target. And the target is the sequence, the 10,000 base pair sequence that contain the switch. So can we cut this thing out? The answer is that we can. And if we do this in human erythroblasts, what happens is that these cells that normally do not contain fetal hemoglobin become fetal cells. So they start synthesizing fetal hemoglobin as a result of the deletions that we have introduced in the genome. We do this also in the cells that we use for gene therapy, in the cells that we transplant in patients. So by doing exactly the same kind of trick, pieces of DNA transfected into human stem cells. When these cells are differentiated into erythroblasts, they become, well, let's say, a good fraction of them become F cells. So become cells that synthesize fetal hemoglobin and start producing fetal hemoglobin chains. I know that this are complicated graphs, but really this is the message. And the message is here. A normal adult stem cells give rise to erythroblasts that produce essentially adult and very little fetal hemoglobin. This is the result of gene editing, 50-50. So we are essentially reproducing a natural mutation that it's hereditary persistence of fetal hemoglobin. And if we do this in cells that come from a patient, this is one of the patients that is followed in a care hospital for gene therapy, for classical gene therapy. If we take cells from these patients and we do the gene editing, not only we do the same, so we start having cells that synthesize hemoglobin F. These are patient cells. These are cells that come from a sickle cell anemia patient. Again, we switch the balance between the bad hemoglobin and the good hemoglobin into the cells. And by sort of simple in vitro test that monitor under a special type of microscope that allows us to follow the sickling phenomena, so the change of shape that these cells have when they are from sickle cell disease patients. Essentially, we reduce the sickling phenomenon by producing hemoglobin F into these cells by gene editing. So this is feasible. It is, well, we are, of course, far from proposing this directly in patients now, but these experiments show clearly that we can induce hemoglobin synthesis, fetal hemoglobin synthesis in adult cells. We can induce fetal hemoglobin synthesis in cells from patients with sickle cell disease. And the result is potentially therapeutic. Now, needless to say, around gene editing, there is an enormous amount of economic interest. Here you just see some of the biotech that developed around editing technology, Sangamo around the zinc finger technology, Selectis around another version of designer nucleases that are called talents. And for CRISPR-Cas9, there is a sort of a nuclear war between these two companies. One is CRISPR Therapeutics and the other one is Editas. And the reason is patents, of course. So the future will necessarily pass through the industry of this type of technology. And of course, now we go back to the reason for Genopole for existing. So the natural environment for developing these kind of things is the sort of environment that Pierre conceived many years ago. So a place where industry and scientists can talk to each other and create innovation. I would like to end my presentation with a couple of considerations, just maybe you can find interesting. So now we have two forms of gene therapy potentially available. A classical one, modified viruses with genes. It's complex, expensive, but it works. We know it works. We know it's safe, and we know it works. And then the new kid, which is direct genome editing. How do we choose between the two? And what are the differences? Well, we don't know. But there are things, there are considerations that we have to do. First of all is efficiency. Are we efficient enough in doing our gene modifications to propose this for a therapeutic approach? Probably not. Gene therapy in 25 years became a very efficient and safe technology. It will be difficult for gene editing to get to the same point. The bar is very high now. Second, there are things that we can correct in the genome, for instance, mutations, but there are things that we cannot correct in the genome. If there is a large deletion, if there is a complete piece of DNA that is missing, we cannot correct this by gene editing. So there are mutations in which the solution will continue to be adding a copy of a gene and not correcting the genome. And then, of course, there is the issue of safety. after many years in which we learned that viruses can actually do bad things in our genome, we now have a technology that is relatively safe for classical gene therapy. What do we know on gene editing? Well, we don't know very much. Well, we know that, of course, that the molecules that we design theoretically go only in one place in the genome, choose one sequence exactly. But there is nothing exact in biology. So these molecules can make mistakes. They can do what we call off-target effects. So they can go randomly in other places of the genome that maybe are similar to the sequences that we want to target, but not identical. Is this dangerous? We don't know. And anyway, if the cell does not repair well enough the sequence that we cut, what are the consequences? We don't know either. The tests that we have are in vitro. We can test things in cells, in culture. But of course, we need to test this into more complex organism. And another things that we learned in gene therapy is that animals don't always reproduce the complex animal that is the human being. And I'm afraid that we will have clinical trials to explain and to prove us that this technology is safe enough. People, of course, is doing their best to improve these molecules, to make them even more and more error-proof. But as I said, nothing is perfect in biology, and we will have to learn the hard way by just trying to do it. Let me just thank some of the people, just very few of the people that contributed into the work that I presented, and others we are doing at Généthon, the team in Généthon, and in the team at Imagine, and Narita Mitchell in particular, and our collaborators in CRISPR Therapeutics and somewhere else in the world. And I cannot finish without acknowledging, of course, the source of most of the money that we use to do this, waiting for the Industrial Revolution, and this is Généthon. Thank you very much for your attention.
Merci. Merci beaucoup, Fulvio, pour cette présentation qui nous a replacés dans l'extraordinaire aventure effectivement du téléthon et de la thérapie génique. Je pense que nous avons des petits problèmes de liaison de micro. Voilà. On va essayer celui-là. Merci beaucoup. Et qui nous a exposé effectivement l'importance, notamment pour vous et tant d'autres, de nouvelles technologies, en tout cas de nouveaux outils, comme le CRISPR-Cas9, dont vous avez longuement parlé. Merci beaucoup pour le micro. Alors, y a-t-il une ou plusieurs questions à Fulvio maintenant?
Qui je peux essayer de répondre en français.
Voilà, c'est pour ça que j'ai repris en français, parce qu'il est modeste, il parle quand même très bien français. Y a-t-il des questions? Peut-être en attendant une question, un mot peut-être sur les aspects éthiques, puisqu'on va parler, et ça nous ferait une transition dans un instant d'éthique avec Hervé Steinweiss. Est-ce qu'aujourd'hui, on n'a pas des vraies préoccupations, vous comme scientifique, chercheur, est-ce que la question éthique de l'utilisation de ces outils comme le CRISPR, dont on dit beaucoup de choses, y compris qu'elle pourrait faciliter énormément d'actes qui pourraient être ensuite problématiques pour l'humain, est-ce que vous prenez en compte cette question éthique?
Oui, certainement. Bien évidemment, à chaque fois on a une nouvelle technologie, il y a un problème éthique qui se pose. Donc, je me souviens des années 80, en fait, beaucoup avant, les années 70, quand il y avait la technologie qu'on s'appelait de l'ADN recombinant, donc qui aujourd'hui on appelle biologie moléculaire, tout simplement. Et il y avait déjà des questions éthiques. Qu'est-ce qu'on fait si on commence à faire des choses avec l'ADN? Est-ce qu'on va créer des organismes qui vont détruire la planète?
C'est la conférence d'Asilomar, notamment.
Oui, exactement. Donc, et au plan médical, évidemment, aussi, je me souviens, quand la thérapie génique a démarré, il y avait exactement le même type de questions. Est-ce que là, avec la thérapie génique, on peut créer des superhumaines ou des subhumaines, hein, ça dépend, ou faire des choses qui, au plan éthique, ne sont pas raisonnables. Évidemment, oui, ça c'est vrai avec toutes les technologies qu'on a à disposition. Avec CRISPR-Cas9, évidemment, on peut imaginer de modifier un embryon, on peut imaginer de modifier des cellules de la lignée germinale, et donc de créer des mutations qui soient transmises à la génération suivante. Donc, ce n'est pas une thérapie génique, mais c'est une modification permanente du patrimoine génétique de l'être humain. Est-ce que ça est éthiquement raisonnable ou pas? Aujourd'hui, la réponse probablement de tous les scientifiques est non. On n'a pas l'intérêt à modifier la diversité génétique de l'être humain. Mais on ne sait rien du tout de ce qui va se passer il y a 100 ans. Peut-être que les générations futures vont trouver éthiquement possible de faire des modifications qu'aujourd'hui, nous, on pense être au plan éthique pas vraiment raisonnable. Mais il y a déjà des gens qui travaillent là-dessus. On sait déjà qu'on peut modifier le patrimoine génétique d'un embryon. C'est tout à fait possible.
Voilà, c'est une des grandes questions qu'on abordera notamment avec Hereditary à la reprise après la pause. Merci infiniment. Fulvio Mavilio, on peut l'applaudir. Merci beaucoup. Et donc, comme promis, une brève pause, d'autant qu'il y en aura une autre juste avant l'intervention de Pierre Tambourin. Donc, merci infiniment, vu le retard, de jouer le jeu, d'autant que les officiels qui viendront pour remettre la Légion d'honneur n'auront pas, eux, possibilité d'être en retard. Donc, merci beaucoup. 10 minutes, pas plus. À tout de suite. Merci.